Hi guys.
Hey, Sonja.
Hi.
I have a problem, and it's about the bioinformatics classes we had the last three times in class, and I didn't get anything because everything was in Spanish. Can you please help me?
Sure.
Yeah, what didn't you get?
Well, I can tell you what I got so far, and then you continue maybe.
Go ahead, girl.
Okay, perfect. Well, what I got so far is what is bioinformatics, and in the easiest way, I would say that bioinformaticians just use computers to analyze biological data, right? And if we analyze this a little bit further, like the name already reveals, bioinformatics combines biology with computers, and with biology, we usually mean biological data or biological information like genes, genomes, proteins, cell, or also ecological system, and so on. And the computers are used to analyze and interpret these data, find patterns or oddities in them, and with the found information, the bioinformaticians try to predict a future outcome or make a model that best describes the biological system, or simply just find an equation that represents the biological system. And as you can imagine, for all of this is not sufficient to just have a computer. Statistics is a huge part of bioinformatics. As you can see in the picture, the best bioinformatician is a mixture between computational science, statistics, and biology. And what is also very important is programming or mathematics or chemistry. And this is the reason why it is a very interdisciplinary science, bioinformatics, as you can also see in the picture. And why is it important? Well, so as far as I got, it is very important, or many scientists say that nowadays bioinformatics is getting more and more important because back in the days, the problem in biology was to get information, for example, to sequence a gene. Nowadays, this is not a problem anymore. This is fast and easy because of all the high throughput methods. But the problem nowadays is to deal with all the information that we got from sequencing, for example, and what does it mean? And for this, we need computers, and that's why bioinformatics is very, very important, or is getting more and more important nowadays. And this is actually all I have in my notes. But the next thing he mentioned was something about software tools. Did you maybe get something about that?
Of course, we can explain it, right?
Yes, and yeah, don't worry.
Well, there are systems or software platforms that can relate and compare databases without any kind of programming or coding. They facilitate like DNA sequence analysis, or they provide an easy conversion of a vast range of data types. Also, the analysis of data, including genome browser or SNP calling and RNA sequence expression, and analysis, and many other things. The main examples are CLC, Genius, and Gene Codes Corporation. Vane, can you explain, Sonia, what all of these tools can do?
Sure. So first, CLC Bio is the leading bioinformatics analysis and visualizing platform, providing seamlessly integrating desktop and server software, optimized for best performance. Now we have another tool that's called Genius. This one is the one that is the world leading bioinformatics software platforms. This has been specially designed and purpose-built as an enterprise solution for commercial antibody discovery and screening. Now, the last one that the teacher explained us was Gene Codes Corporation. This is another international software. This one is specialized in being scientist-friendly software tools that hold the dominant share in the DNA sequencing and are unrivaled in forensic DNA identification for mass fatalities.
Okay, but what is sequencing and why is it important?
Well, first you need to know that there are many differences between DNA and RNA, so that you can sequence a genome. Like, for example, DNA, it
has both have sugar, but DNA has deoxyribose, and RNA has ribose.
And also the base pairs or nucleotides that each one has is that DNA has thymine instead of uracil, like DNA does.
Yeah, and DNA is double-stranded, RNA is single-stranded. And about the stability, DNA is more stable than RNA.
Well, you can do sequencing from DNA or also from RNA, but you need to take it like from the messenger RNA and to cDNA and then start from there.
Because sequencing is the process of determining the precise order of the nucleotides in a DNA molecule.
Oh, and it is important because it is basic for biological research and in many applied fields, such as
Medical diagnosis, biotechnology, forensic biology, virology, and biological systematics, and well, so many.
so far so good.
Ah, okay. Now I got what sequencing is. But now I have the sequence of a gene, and it's on my computer. What can I do with it right now?
Well, many things, but the principal ones are that it allows scientists to identify changes in genes or
Associations with diseases and phenotypes, and also identify potential drug targets.
and study like how different organisms are related and how they evolved. One of the examples that we saw in class for technologies for sequencing in large scale is Illumina, that involves fluorescent nucleotides, and it is actually a very complex process, but
Actually, there is a video, so we can send you the link and you can check it.
it is very interesting. You will learn a lot. And I remember that the teacher said something about looking for genes
And the steps that they are involved.
yeah, but I can't remember many of it. Maybe we should call Solange and Rogie.
Yeah, that's a great idea.
Hey, I have an income call. What do you need?
Hi,
Hi.
Rogie. Hi, Solange.
We are helping Sonia with the bioinformatics class, but we have some doubts about the gene searching and that stuff.
The steps that we need to follow. So, can you explain us about it, please?
Yeah, of course, we're actually experts, right, Solange?
Yes.
So why don't you just tell us?
In general, when we better know a system, we can modify it in a faster and more efficient way. Bioinformatics is in charge of storing and analyzing and interpreting enormous amount of results obtained of the DNA sequences. This process is summarized in three major stages: sequence identification, sequence, and functional annotation.
Okay, guys, so I have my computer here, and finding a gene is actually very, very easy. You're just gonna have to follow these steps. First, you're gonna type NCBI on your browser. Then, you're gonna click on National Center for Biotechnology Information. Let's say you wanna find the gene. Then, you're gonna find a filter, and you're gonna go to the gene section. Then, you type the name of the gene or the code if you have it. In this case, let's just say I wanna study the KRAS sequence from oncogenes. So I just type oncogenes and click search, and I have the first one, Homo sapiens KRAS. I have to click on that one, and let's say if I wanna analyze that gene, first it's gonna give me all the data about the gene. And then it's going to give me a series of sequences. The first one is going to be the complete sequence, but it's going to have introns because it's only RNA. And then we're going to have like the different ways in where we can search parts of the gene to study it.
And there are another databases that allows you to complete your investigation, like IDT. You can explain, Rogie?
Yes, IDT is the oligo analyzer tool, and basically it gives you different sets of primers for your. Let's say you wanna isolate this gene KRAS again. You need to have like the best primers with with the highest level of fidelity for your study. So oligoanalyzer is gonna give you a set of design primers that you can use to isolate this one, and it's gonna give you like 10 options, and it's gonna be like this one is more resistant to heat. This one is better because it doesn't brine this thing, and yeah, that's it.
Ah, okay. What was the name again? IDT. And this is the website. You open it and you just put inside your gene, the name of your gene, and then it will give you like a list of primers that you can use, or and then you can choose, or what?
Exactly, Sonia. Once you're in IDT oligo analyzer, what you're gonna get is well, you put your sequence there. You're gonna get a list of primer sequences that you can choose from to do your research. There are, as we learn in our genetic engineering classes, there are different factors that can affect the fidelity of your primers, and you need to evaluate these factors in the following sequences that you're gonna get. And actually, you can click on the sequence and they send it to you. So that's what they make. That's what they really do. Like they sell primers to you.
If you are looking for the structure of your protein, you can use different databases like PDB. In these databases, you can find the three-dimensional structure of proteins and nucleic acids. Or SIB is a non-profit academic foundation that promotes research and development of database in the fields of genomics, proteomics, and systems biology. And if you are looking for domains and motifs, you can use ProCite. This database offers tools for the analysis of protein sequence and detection of protein motifs. Or UniProt is freely accessible database of protein. That's it. It contains a large amount of information about biological function of proteins from the literature.
Okay, this works if you know like which genes you're working with. But do you know what happens if we don't know the gene? Like if we have something but we don't know which gene is?
Really, I don't know.
The professor says something about phylogenetics or some stuff like that.
Right. We should.
Yeah, we should ask somebody else. Like
Yes.
I think Miguel knows.
Hi. You were mentioning before about a question about phylogenetics. Basically, phylogenetics is the study of the evolutionary timeline of each living being and also for certain proteins and genetic sequences. So, mm, the main point of phylogenetics on bioinformatics is because when you have a gene, but you want to check also for any gene that may be similar, you use an algorithm in order to check which will be the best candidates. So, for example, on this case, we got what will be the UPGMA method and also the neighbor joining method. Those were the first methods created in order to check for the candidates on each gene. For example, for the UPGMA, which is the unpair group method with arithmetic mean, was created around 1958, and it was focused on checking for the protein evolution. The disadvantage of this method is that it didn't take into account the time that it takes from point A to point B. Even when those two candidates are close, the time that they take for them to evolve was different, so it got this disadvantage. While the neighborhood joining takes that you have a branch and the branch got its leaves. So the leaves that are closer are a neighbor, and those are inside a cluster. So you get several neighbors in order to build a whole cluster, but that's taking into account the time that each molecule took for evolving, and the ones that are closer be, because of their similarities, also genetically speaking. The important thing of this is that maybe you have the gene that you're looking for, but it didn't quite fit of what you were actually expecting. So you wanna try something different, but that is similar. Well, finally, it got the advantage that you can select, that you can try several um, candidate at once, more with the neighborhood joining rather than the UPGMA.
Okay, that was great.
And what is all this for?
And yeah, can you tell us some applications of this?
Well, for applications, I don't know if you heard about that, but on 2015, Elon Musk and also Craig Venter were involved on a really brilliant idea of you want to go to Mars. Okay, yes, but the planet is not suited to sustain life. So how are you going to transfer the necessary elements to transforming that planet to sustain life? That process is called terraforming, and actually, well, take several trips just to take samples for bacteria, for plants, in order to start creating life there is not going to work. For example, Elon Musk knows that he's not going to see a colonized Mars or not a fully colonized Mars on his lifetime. But the idea that both of them have is to, instead of shipping the life there, just shipping a machine that can print the genetic codes. This also comes in hand with synthetic biology. When you create a blank organism, and just fill it with the genetic information that you printed with the machine. So you send the instructions from Earth that you get through bioinformatics, and then you print it using synthetic biology on Mars, and you start creating their bacteria and also more complex organisms like fungi and also plants. So you start creating the environment that we need, and once it's ready, you can send then persons to start colonizing the planet. So it's a better process than just having several trips trying to send a fully created organism from Earth and seeing that it's not working because it doesn't have the necessary conditions yet. So it's actually quite neat.
Well, thank you so much, Guy. I got a lot more than before because I didn't know anything. So thank you so much. You're the best classmates ever.
Wow, each one of us is essential for this team.
We are the best.
Solange, I'm so proud I'm gonna die.
We are a very interdisciplinary team.
So interdisciplinary.